The second speaker on this is David Boulware from Minnesota, and he's going to talk on Uganda cryptococcal trials on oral amphotericin. Now, what you've seen with Tom is he took drugs that are already there and tried to make them and use them better. I think what David's going to talk about is another step, which is maybe new drugs to actually study and improve the outcome of Cryptococcus.
Great. Thank you very much, John, and it's a pleasure to be here. I'm going to talk a little bit about kind of the story, a little bit about how we got to where we're at and some of the new work we're doing with an oral amphotericin compound. And the concept of an orally available, non-toxic formulation of amphotericin sounds, like the holy grail, sounds too good to be true. And so I'll show you some data that we have from our ongoing phase 2 trial. So this is sort of the Uganda crypto studies. And so our clinical trials have been funded by a variety of sources. This phase 2 is funded by NINDS, and it's in collaboration with Matinas. We've also had a prior collaboration with Amplex before they no longer exist, and I'm on a scientific advisory board. And so first off, I might just sort of start with a map of Africa of where is Uganda for those that might be geographically challenged. And so this collaboration started back in sort of 2003. At the time, this is pre-PEPFAR, pre-Global Fund. There were no HIV medicines. There was no HIV clinic building, but there was a lot of HIV and a lot of interest in HIV care. And so the IDSA and other groups had an IDSA HIV trainers program. And for some odd reason, my mentor volunteered to go as a HIV trainer back in 2003. And that really sort of started a collaboration with, working at Malago Hospital, which this is what it still looks like, and with the Infectious Disease Institute that opened in 2005. And with this is really a long-term collaboration, if my slides continue, in partnership with David May, who's here today at the meeting, as well as with Andrew Kambugu. And so why did we look at meningitis and why particularly Cryptococcus? And so what I learned in med school, and I think probably everyone else did, was that there was the meningitis belt in Africa, and this is all meningococcal disease. And it's, certainly that was true prior to the rollout of vaccination. But then there's sort of sub-Saharan Africa. And within sub-Saharan Africa, HIV exists and really dominates what the etiologies are of meningitis. And so if you look across sort of a number of studies conducted in the last decade, you'll see that the most common etiology is Cryptococcus. Second most common probably is tuberculosis, if you could actually diagnose it. And so in Uganda, Cryptococcus is more common than all other causes of meningitis put together combined. And so obviously driven by HIV. And so this was published in 2013, this table, but it hasn't changed. And so in last year, it was 60%. And so we took the plunge on clinical research back in 2006. And this might be a good time to mention that there is the International Cryptococcus and Cryptococcosis Conference that's being held in January. There's a lot of great things about the conference as well as about Uganda. And so rafting on the Nile River is one of those. And sometimes, with clinical research, everything goes, nice and smooth and swimmingly well. And other times, it's a little bit more challenging. And so within our research, we kind of asked three fundamental questions, I think, is what our research team does. The first is how best to diagnose meningitis. Why do people die of meningitis, and how to prevent those deaths? And so very simple questions, but at times sort of overwhelming and sort of, there's a lot of work to be done. And so when we started back in sort of 2001, 2003, this is work with Andrew Kambugu and others. When we looked at 2 weeks of amphotericin deoxycholate among people with normal mental status who could consent, the 2-week mortality was 50%. And so that's where we started. And so once people got discharged from the hospital and would start the HIV therapy, they're long since, the majority of people are long since dead. And you can see this is sort of a very, precipitous drop in mortality over that 2-week period. And, first we sort of worked, over the first, 5 years or so really improving survival really through supportive care. So IV fluid management, potassium, electrolytes, monitoring and supplementation. And so we had no new wonder drugs. We're still using IV amphotericin, but we were able to improve the survival with just better toxicity management of amphotericin. The patients were just as sick. The CD4 counts were just as low, but we were able to provide better supportive care. And so amphotericin can be a very toxic drug, but it also can be very life-saving. And these results are similar to the EnACT trial. If you look at sort of 30-day mortality in a lot of those arms, among those with normal mental status, fairly similar levels of mortality. So the 5-year survival is actually pretty good if you can survive initially. And so that's the problem in this initial time period. In the first 18 weeks, 95% of the mortality occurs through 5 years. And so the question is really, what can we do about this? And so better induction therapies, better initial treatments, how can we improve the survival? And so this is with primarily amphotericin and fluconazole-based regimens. And so there's a number of different strategies to do this. And so, as I mentioned, improving supportive care, optimizing the pharmacokinetics, and so what Tom Harrison just presented with the Ambition trial, and sort of optimizing PK of drug delivery is one aspect of that. Repurposing existing drugs, and so we tried that. Didn't quite work out as we wanted to. And then there's new drugs. And so sometimes things don't work out exactly how you want to. And so some of the new drugs that we've been working on is encochleated amphotericin. And so I'll refer to this as CAM-B. It's also MAT2203, and it's made by Martinez. And Martinez is going to talk, I believe, tomorrow night at dinner and talk a little bit more about the compound. And so there's a number of sort of milestones that have been completed. And so Peter Williamson, who's here, did a lot of the preclinical work in mouse models, and that's published, showing that the cochleated amphotericin can get into the brain and is co-located with with macrophages, monocytes, and with Cryptococcus yeasts. We did our phase 1 study, which I'll show, and then our phase 2 study, which is ongoing and should be finished hopefully by the end of the year. And then we have a phase 3 trial that will start up hopefully next January. So when you look at sort of the PK in humans, some of the prior work that was unpublished, initial sort of standard sort of dose escalation studies for single ascending dose. As you increase the dose, you got sort of less increased absorption, and you had more GI symptoms. And so when we looked at these data, this sort of basically the symptoms were driven by malabsorption. And so what I kind of make the analogy back to is sort of the Olestra potato chips back in the 1990s, where the non-absorbed fat, that if you ate a whole bunch of them all at once, you get sort of bloating and sort of nausea, and you didn't feel very good, and maybe some loose stools. And so it wasn't going to kill you, but it was a little bit unpleasant. And so those potato chips are not, I don't think they're allowed in Europe, and they're not as common as they used to be. But that's kind of the type of symptoms. You're getting sort of non-absorbed fat, and so you can get some GI symptoms. It's sort of mild, not particularly distressing, but certainly present if the dose is too high. And so we had a hypothesis that if we basically separated out the dose and did multiple smaller dosing per day, we could increase the total dose received with sort of minimal GI symptoms. And so we started a phase 1 study. I think, if I hit the right button. Basically, we did sort of three doses of sort of 250 4 times a day, 300 5 times a day, and then 333 6 times a day for 2 grams a day, and basically looked at GI symptoms. And so it was sort of an initial sort of single daily dosing, and then we did sort of a week-long dosing. And so within this study, basically we were able to get up to 2 grams a day. At 2 grams a day, there were, some side effects, but in moving forward the dose, we wanted to use the sort of the maximal tolerated dose that was possible, because if we used the maximal tolerated dose and it didn't work, then we knew the compound wouldn't work. Versus if we took, let's take a lower dose, but no one will have any side effects, but if it doesn't work, then at the end of the day, do you come back and say, well, what if we picked a higher dose? And so we wanted to go with sort of the highest tolerable dose into our phase 2 trial. And so the phase 1 trial was led by David Maya. That's a picture from yesterday of some fishing. He caught his first fish. And really, the study coordinator, Adkunda Mukazungi, has really been great for this study. And Caleb Skipper, who's also here, was highly involved in the phase 1 study. And so for the phase 2, sort of took the baby. There's another picture from Uganda once again. There was a crypto conference in January. Would encourage people to go, but sort of the baby study sort of launched into more the full phase 2 trial. And this is our study design. So it's kind of complicated. And I think FDA said, well, it's really complicated, but we do like it. And so basically there was a kind of 4-stage design, and we're in the last stage now. But the first stage was just seeing, is the dose tolerable? And so we started out by giving people 5 days of IV Amphotericin, which has been done before, but with good outcomes, and then switching to the oral regimen. And so that regimen basically didn't depend on the oral drug to do anything, but just was it tolerable? And so in that first cohort, we realized, maybe the dose was a little bit high, and so we backed off from 2 grams a day to 1.8 grams a day in the second cohort. The second cohort really was based on efficacy of doing 2 IV loading doses and then switching to oral therapy. So as with many things in medicine, we often use loading doses before sort of using sort of maintenance or switching to oral. And so can you switch early to oral dosing? Stage 3 was kind of a little bit of a safety cohort of just 10 people with experimental regimen of giving them the drug for 5 days and then switching to sort of scheduled rescue therapy with IV Amphotericin, not knowing, did you need loading doses? How would people do with this? And the concern of, before we threw people off a cliff with a drug that may not be well absorbed and have a bunch of fatalities, that we would at least have sort of a scheduled rescue therapy built in. And then the last stage is an all-oral stage where they start on oral therapy, and it's sort of in comparison with what the WHO standard care is of 1 week of IV Ampho and 5-FC. And so within this, the primary trial endpoint was early fungicidal activity, and so this is piloted by Tom Harrison's group initially, and we've done it as well. And so because you're doing multiple longitudinal LPs to control, therapeutic LPs to control pressure, you can also do a quantitative culture, and then you can basically plot what the rate of clearance is. And so that's generally approximately linear on a log scale, and you can basically quantify what's the rate of clearance. And this is fairly unique because it's antifungal activity in humans at the site of infection. And so for most things in ID, we really can't do that, but here we can. And so this is very nice for phase 2 trials to get an idea on what the activity is. And so this has been used in multiple trials, as Tom mentioned before, with higher dose fluconazole, with Ampho Fungicidal regimens. And when we've looked at this in our own cohorts across multiple studies, this is not rocket science, but if you have poor CSF yeast clearance, you have higher mortality. And so in particular, it's really if you have bad CSF yeast clearance, and so basically below this sort of threshold of 0.2 CFUs per milliliter of CSF per day or worse, the mortality really jumped up. And so as long as you're above that in our prior cohorts, people had fairly similar survival outcomes. And so, as I mentioned, we had tweaked the dose down for the phase 2 cohort, and this study design where basically 2 days of IV loading dose and then switching. This ran sort of during COVID, but during sort of the first half of 2021. And so when we look at this rate of clearance, this sort of early fungicidal activity, probably the best comparison is, or the most robust comparison is in the light green, which is of course the hardest to see. And so that's the ambition sort of standard of care. And so that's the sort of mean estimate among 371 people who are randomized to the control group. And so the other groups on stage 1, 2, and the controls, they're a little bit higher, a little bit lower. Don't get too excited about that because they're smaller subgroups. But as you can see, that the last group there is the oral, the stage 2 cohort where there were 2 IV loading doses of Ampho, followed by the oral Can-B, and the rate of clearance overall is pretty similar. And so we were pretty reassured by this, and this we thought was quite exciting that we're seeing very similar levels of activity with this. But of course, that's nice, but what about survival? And so impressively, in the survival in the stage 2 cohort, our 18-week survival was 90%. And so that was better, that was not statistically better than the IV Ampho controls. And if you look in the first 30 days, there was only one death, and that occurred on day 1, actually before they even received the drug. And so the 30-day survival was 39 out of 40 participants. And so we think this is pretty good. So if you go back to my first, Kaplan-Meier curve of the 50% 2-week mortality where we started, getting a 90% 18-week survival is sort of just beyond our wildest expectations. And so we're pretty happy with this. This is a cohort that does start with normal mental status. They have to be able to take oral medicines, but they still have a CD4 count of 30. They're still a pretty sick cohort overall. Some of the other results which are of interest, Sterling was pretty good. There was a little bit higher rates of the renal insufficiency in this cohort, but otherwise all the rates of adverse events were lower. And so looking at anemia, looking at other factors were lower. And so the creatinine was a little bit anomalous. And so this, I think we learned after the stage 2, was just because you give sort of IV Ampho for 2 days and then you switch them to an oral drug, some hospitalized patients still are pretty sick and need some IV fluids for a couple days more. And so on an ad hoc, standard sort of basis, we've been doing this in the latter cohorts 3 and 4, and we haven't seen any further sort of renal toxicity. And so just noting that if just because you're on an oral therapy, you still have to take care of the patient and sort of pay attention to the big picture stuff. And then I'm going to skip through I think the rest of. I have one more slide here. And so, what about the rates of GI side effects? And so overall, yes, it's higher than not giving an oral medicine, but the vast majority of people either had no days of any side effects, so 45%, or sort of 1 day of any side effect, or 2 days. And so there were a couple people who had more, if you look at nausea, vomiting, diarrhea, had a few more rates of GI side effects, but nothing too outlandish overall. All right, so I'm going to stop there because the remainder of my slides, I realized I was told I shouldn't present because it's ongoing data that hasn't been publicly revealed. But our cohort 3 and 4, so Matinas is a publicly traded company. As an academic person, I'm not always very clued up on SEC rules for public disclosure, but the cohorts 3 and 4 are sort of ongoing. We've got about another 10, 11 patients to recruit. Hopefully we'll be done sort of in the fourth quarter of this year. And so we're overall sort of pleased with how the remainder of the cohorts are going and planning to move forward with a phase 3 study that's going to be hopefully funded by NINDS come early 2023. So I'll stop there.